All right, so this is metabotropic receptors, and this is where the video kind of cuts off. So what I'm going to do is I'm going to try to keep it under 15 minutes so I can post it to YouTube, and then we can kind of go around the whole Echo 360 debacle. So the first thing we want to talk about when we're talking about metabotropic receptors is the fact that the response of the receptor, or the response of the neuron to binding of a ligand is entirely dictated by the identity of the alpha subunit, so by the identity of the alpha subunit of the heterotrimeric G protein complex. So that's the hetero for different, trimeric, which is 3 different subunits, G protein complex. So what do I mean by this? Well, when we have a G protein coupled receptor, let's say this is a cell, and we have our 7-pass, I think I did more than that, so 1, 2, 3, 4, 5, 6, 7, our 7-pass transmembrane regions, we have our G protein complex right here. And it's heterotrimeric because it contains the alpha, the beta, and the gamma subunit, which are all different. And the alpha subunit is conjugated to a molecule of GDP. The identity of this alpha subunit is going to be what determines the overall effect of the entire structure, meaning you could completely change the identity of the receptor to bind to a different ligand, and the response would be the same because all it does is convert this GDP or exchange this GDP with a GTP, which activates the alpha subunit. So that's important to keep in mind going forward. It's always about the alpha subunit of the G protein coupled receptor. In addition to that, they're all named exactly how they should be. They're named for the effect of the alpha subunit on the entire pathway. So this is stimulatory, and this is the alpha subunit, right? So a G alpha S is going to have a stimulatory alpha subunit, whereas a G alpha I is going to have an inhibitory alpha subunit. And we'll get into a little bit weirder ones later. But what I want to do is I want to start with G alpha S, right? So that's going to be the stimulatory G protein coupled receptor pathway that's dictated by the identity of that stimulatory alpha subunit. So if we take our cell here, we can draw, and I'll draw all this in green so we know so we remember it's stimulatory. We can draw our G alpha S. So here's our G protein coupled receptor. It has 7 transmembrane regions, and it's conjugated to a heterotrimeric G protein complex. The identity of this alpha subunit is going to be alpha S, so stimulatory. And before any ligand binds, it's going to be conjugated to a GDP, which is just the diphosphate form of a GTP, right? So once a ligand binds to this G protein coupled receptor, this GDP is going to be swapped out for a GTP, which is going to activate the alpha subunit. And it's important to remember that these subunits actually always stay associated with the membrane because they're peripheral phospholipid bilayer proteins. But what's going to happen is this GTP is going to activate this alpha subunit, and this beta and gamma are just going to kind of go off to the side. As far as I know, they don't do much in the G alpha S pathway. I'm sure they do something, but it's not really important for us to know. The major effect of this alpha subunit being active is going to be the increase in, or the positive regulation of a protein known as Adenylyl Cyclase. And I'm going to draw this in black because it's affected by 2 different pathways. So Adenylyl Cyclase is a 12-pass transmembrane protein, right? So it's going to be conjugated to the membrane as well. Adenylyl Cyclase. So 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. It's not super important that you know that. In fact, I wouldn't care too much about it. But one thing you should know is that the role of this Adenylyl Cyclase is to convert ATP into ADP. Convert ATP into cyclic AMP. So the effect of this is going to be ATP to cyclic AMP. So what the alpha subunit of the G-alpha S pathway is doing is it's upregulating the effect of this adenylyl cyclase. It's upregulating the function of this adenylyl cyclase to lead to an increase in CAMP expression. So increase cyclic AMP expression. And I'll draw this arrow coming from adenylyl cyclase. Right. So you get increased CAMP expression. What that eventually leads to is an increase in the activity of a protein known as protein kinase A. And I drew it in black because, of course, it's going to be affected by another G protein coupled receptor pathway. So, right, we can tell that the major effect of this stimulatory G-alpha S heterotrimeric G protein complex is going to be an increase in activity of pKa. The major effect of that is going to be most likely the phosphorylation of some type of channel to allow in ions. And you might be asking yourself, why would it go through all of this convoluted mechanism just to get back to opening an ion channel? And the reason for that is it's all about signal expansion or signal amplification. So not only does pKa most likely, let's say it opens a sodium channel to allow current in, it most likely also does a whole wide range of other stuff. It could lead to transcriptional changes. It can lead to a whole manner of things that we're not going to talk about. But just remember the function of this pKa in a sense of signal amplification. So for our cases, we're just going to focus on pKa itself, right? All we care about is that pKa becomes active, and it leads to stimulatory events. So I can draw here stimulatory events. That's going to be influx of current, stuff like that. But we have another G protein coupled receptor pathway, or another type of heterotrimeric G protein complex that's going to do the opposite of this. And this is going to be that G alpha I, sometimes you see it as G alpha IO, but I would just focus on the I, right? The I stands for inhibitory. So you're going to get a very similar looking mechanism with one very key difference. So here's your 7-pass G protein coupled receptor. Here's going to be your alpha subunit, which I always draw as a triangle, your beta, and your gamma subunit. And there's actually going to be another significant difference here, but what's going to happen is basically the same thing. You're going to get this GDP-bound alpha subunit to be converted to a GTP-bound alpha subunit. And that's going to be in response to some kind of ligand binding to this G protein coupled receptor and leading to this exchanging effect of the GPCR. So once this is active, once this alpha subunit is active, it's actually going to do the exact opposite of the G-alpha S. So it's going to inhibit the effect of adenylyl cyclase, right? And that's going to lead to a decrease in cyclic AMP production and a decrease in pKa activity, which is going to lead to or be associated with more inhibitory events. And one thing I want to make clear, just because I forgot to mention it at first, metabotropic receptor equals G protein coupled receptor. A metabotropic receptor is a receptor that's coupled to a G protein. So that's just terminology for you. Just remember that those two are synonymous. In addition to this inhibition of pKa, the beta and the gamma subunit actually have a role here. So what they're going to end up doing is they're going to open what's known as a GERK channel. And this is important to pay attention to because we accidentally said this wrong in class. What a GERK channel is it's a potassium channel that leads to efflux of potassium. So this is going to, if we were to draw a voltage graph, an efflux of positive charge is going to lead to a hyperpolarization. So if this is your resting membrane potential, as positive moves out, you're going to get a more negative membrane potential. And that's going to be, again, inhibitory. So that's going to be hyperpolarizing. So that's your G-alpha S and your G-alpha I. And then we have another one that's a little more unique, so we kind of talk about it on its own. But if we were to draw this, we'll just draw another cell. It's a little more simple. But this is going to be your, I'll draw it in regular color. So this is going to be your G alpha Q pathway. And this honestly can lead to both excitation and inhibition in different cases. So we're just going to talk about kind of the mechanism for it. But what it is, right, it's same 7' G protein coupled receptor, but it's coupled to an alpha and a beta and a gamma, in which the alpha subunit is this type Q. And it's, again, it's not stimulatory or inhibitory. It's kind of more related to transcriptional changes and more synaptic modulation type effects. So what's going to happen, and you guys don't need this spiel more than twice, so I'll just say that ligand binds leads to activation of the alpha subunit. So here's your alpha subunit, right? It's going to be bound to GTP, and now it's activated. What it's going to do, and it's not going to affect adenylyl cyclase, it's actually going to affect a different protein known as phospholipase C. Phospholipase C is going to cleave, so it's a lipase, so it's going to cut something. It's going to cleave this bond right here. I'll draw that in a different color. So it's going to cleave this bond right here that exists within this molecule known as PIP2. By cleaving this bond, you get two different resulting molecules or chemicals. So you get IP3, which is an acetyl triphosphate, and you get diacyl glycerol, which is also known as DAG. So cleaving this PIP2 results in DAG and IP3 being produced. DAG, or diacylglycerol, goes on to increase the expression of what's known as protein kinase C, right? So that's in contrast to protein kinase A. Again, this is going to have, a wide range of effects. One of them could be opening an ion channel leading to excitation or inhibition, but it's all about signal amplification in the case of GPCRs, and that's important to remember. So this has a wide range of effects. IP3 is going to go and actually interact with a structure we talk about in a lot of other classes, which is the endoplasmic reticulum. And one important feature of the endoplasmic reticulum is it has a high concentration of calcium ions. So those calcium ions are going to have a driving force to leave the endoplasmic reticulum. So what you get is a binding of this IP3 to a receptor that's located on the ER, so there's your IP3 binding, and that actually leads to calcium release. So this is calcium. Calcium release itself can lead to activation of protein kinase C. So again, that's even more of a signal amplification type event. But it also leads to calcium calmodulin kinase, which leads to a whole manner of things. So again, this can lead to transcriptional changes and a bunch of stuff that we're not going to talk about. But just remember that you're always talking about signal amplification, you're talking about transcriptional changes, you could be talking about a wide range of phosphorylation events, remember, because this is a kinase, so this is going to phosphorylate all kinds of stuff, which could be, an ion channel. And then the last one we're going to talk about as we close up this video is G alpha 12-13, and he mentioned gusdeucin and transducin, which are gusdeucin, transducin. These are other types of alpha subunits. These are going to be involved with taste and vision, respectively. So we're going to talk about those later. You don't need to know them now, but just remember that there's other types out there. G alpha 12-13 is unique. So if this is your dendrite, it has the surface area in this region for, let's say, one synapse to form. So right now you have one synapse to one dendrite. This is pretty strong of a connection, but what you could do is you could actually release factors that bind to what's known as a G alpha 12-13, and the effect of this is going to be more related to mobilizing actin cytoskeletal rearrangement. So the overall effect you get is actually a change in the shape or morphology of the dendrite to form what's called a dendritic spine. And I'm trying to rush here so I can not go past 15 minutes. But essentially what's going to happen is you have an increase in surface area of the dendrite, which allows for the strengthening of that synaptic connection, right? So now you have not only one synapse being formed here, but let's say 2 or 3 or anything like that, and that can strengthen that synapse. So the next video I'll talk about the model of synaptic modulation in Aplysia californica.